How are you, my dear students? Hope everyone is okay and fine. Today, I also starting our new topics, and our new topics is also the previous class. I also told you these topics about these topics. Today, our new topics is the chapter 3. It's also a repeat class and also repeat topics. And today I also explain about the cell division, and our new topics is about the equational division or mitosis. What is the mitosis and the cell division? Actually, different types of cell division occur in all types of organisms of unicellular or multicellular. In the cell division, sometimes it's also happen about the growth body, and another is the reproductive cells. Today I also explain you the how can the growth of body, and I also explain about the mitosis. Here the mitosis, through this process of division and equal cell, eukaryotic cell and prokaryotic cell. Have you any idea? I also explain these topics about chapter 2. An eukaryotic cell divides into two identical daughter cells. In the process, the nucleus and chromosomes are divided into two. First, also the equational division here also divides two parts. First part is nucleus and another part is the chromosome, and it's also called the equational division. And then the two identical daughter cells are produced with the same type of the chromosomes in number, physical and constructical structure features as their mother cell. This is also known as equational division. Mitosis occurs in somatic cells of eukaryotic organisms. And I also explain the phase of the mitosis. Here the 5 phases of the mitosis. The one phase is the karyokinesis and another is the cytokinesis. Karyokinesis. Karyo means nucleus. First structure also that divides into the nucleus. Here I also draw this. This structure is nucleus and the Latin word is karyon. Karyon means nucleus, and that this structure is karyokinesis, and another topic is the cytokinesis. Karyokinesis, it's also the 5 phase. One phase is the prophase, another is the pro-metaphase, third is the metaphase, fourth is the anaphase, and last is the telophase. First, I also explain about the karyokinesis, and first topic is prophase. Please everyone see the book and the stage of prophase. Here the prophase. This is the first stage of mitosis. At the onset of the stage, the nucleus becomes little larger and chromatin fibers start condensing into short, thick and tightly coiled structures called chromosomes. At this stage, chromosome can be seen with a compound microscope. So, every chromosome then divides into two sister chromatids. At the centromere, they remain attached. At the S chromosomes are still tangled. It is difficult to easily count the number of chromosomes in a cell. Here, everyone see the books. Please everyone see. First structure is prophase, and there is the little structure shown, the nucleus. Another structure is chromatid. I also draw the another space. This structure is chromatid, and the middle structure is the centromere, and its another name is the prophase. Second structure, pro-metaphase. Here it's also very detailed topics. Please everyone see the books. Pro-metaphase. Here, early in this stage, spindle apparatus with two poles is developed in plant cells from fiber protein. I also see the spindle apparatus. This structure is the spindle apparatus, and it's also the another topic is the protein. This is the middle plane of the spindle apparatus is called the equator. Here this structure is another name is the equator, and some fibers of the spindle apparatus made of microtubules of cytoskeleton and stretch from one pole to the other. These are called spindle fibers. And this kinetochores in the centromeres of chromosome attached to some fibers of the spindle apparatus. Kinetochores of the centromere. I also draw this. This structure is kinetochore of centromeres and it's also attached with the spindle apparatus. And please everyone see the books. Kinetochores are protein structures assembled on centromeres and they link the chromosomes with mitotic spindles, and this apparatus also link with the chromosomal apparatus or the spindle apparatus. Here, these fibers are generally called traction fibers. Its another name is the spindle fibers, another name is traction fibers. And they are also sometimes called chromosomal fibers as chromosomes are attached with them. Chromosomes then start assembling on the equatorial plane. The nuclear membrane and nucleolus start to disintegrate and disappear. Please everyone see this. This also the nucleolus. And this structure is the nuclear membrane also the disappear. Okay. Now the nuclear membrane and the nucleolus start the disintegrate and disappear. I also repeat this structure and also I also explain about the pro-metaphase. Here this structure is the pro-metaphase. Another structure is metaphase. Meta means middle. Here everyone see this the chromatid also equator stage or middle stage. Here I also explain the metaphase. At the onset of the stage, all the chromosomes assemble at the equator from the two poles. Here I also draw is the one poles and another is the one poles. These also the two poles and it's also the structure of the spindle apparatus. Here the centromere of each chromosome remains on the equator, but the two arms take positions towards the poles. In this stage, the repulsion increases. At the end of the stage, the division of the centromere starts. The nuclear membrane and nucleolus disappear completely. At the last stage, here no any nucleolus and here no any nuclear membrane. At the lastly, they also disappear from this stage. Students, everyone see the books and also see the boards. I also draw first structure is prophase. Here also is the nucleolus, chromatid and centromere and nuclear membrane. Second structure is pro-metaphase. It is also the shortly disappearing the nucleolus and the nuclear membrane. But finally, metaphase, the nucleolus and the nuclear membrane finally disappear. Now, I also explain the next structure is anaphase. Have you learned the word, the anatomy? Yes, the anatomy, these words come from the anaphase. Ana means the breath. Yes, and now I also explain about the anaphase. Here, the sister chromatid become the two sister chromosomes and they are pulled apart. Sister chromatid. Here this structure is the disappear. One structure is the one chromatid and another is the next chromatid, and we also told this structure is the sister chromatid. Everyone see the books. The clipped centromere moves to the pole while the chromatids trail behind. Here I also draw this. First structure is the centromere and second is the chromatids arms. And another structure is the centromere and another arm is the chromatids arm. Here I also explain again. The clipped centromere moves to the pole while the chromatids trail behind. Each chromosome splitting into two moves to the two poles, and so the number of chromosomes remains unchanged. On the centromere in the chromosome, the chromosomes look VLJI, and the another names is the metacentric, submetacentric, acrocentric, and telocentric. I just draw this. First structure is the metacentric. Here also the centromere is the middle and the chromatid to chromatid. It looks like the V-shaped. And another structure is, its another name is metacentric. Submetacentric, I just draw the L shape. This is the centromere and another is the chromosomal arm. This looks like just also the L shape, and another name is submetacentric. Another structure is just look like J shape. It's also the acrocentric. And two chromosomal parts also the linked, and the centromere is middle, just look like the I shape, and it's the another name is the telocentric. Now everyone see the books. At the end of the anaphase stage, the daughter chromosomes are completely pulled apart to the two poles and start their elongation. Students, I also explained the karyokinesis, and the last stage, the karyokinesis, is the telophase. Telophase, here I also draw this, the anaphase, and another last stage is the telophase. Now, everyone see the books. This is the last stage of mitosis and a reversal of the process. Reversal. Here, chromosomes become decondensed and surrounded by new nuclei. They elongate back into thin and long structures called chromatin fibers and form a mesh called the nuclear reticulum. The nucleolus also reappears. The nuclear membrane reappears around the nuclear reticulum. So ultimately, two new nuclei are formed into the two poles. The spindle apparatus. Now, the spindle apparatus disintegrated, and so the spindle fibers gradually disappear. Here I also draw this, the telophase. Here also again the repeat and reappear nuclear membrane. I just draw the nuclear membrane. Another structure is nucleolus and the chromosome, but it's not also chromosome, just look like a chromatin fiber. And there also the cell plate, and then began to the return of the karyokinesis, and the last structure is the cytokinesis. Now I also explain about the cytokinesis. What's the happened of the cytokinesis? Please everyone see the book. Cytokinesis. Here, at the end of the telophase stage, some small parts of the endoplasmic reticulum aggregate in the equatorial plane and collectively form the structure of the cell plate. Is the endoplasmic reticulum is from the chromosomal or the cell plate. Here I also draw this collectively from the structure of the cell plate. Equal distribution of the cytoplasmic organelles is accomplished. As a result, two identical daughter cells are developed. In the case of an animal cell, a contractile ring on the plasma membrane and the equatorial plane pinch of the two nuclei. Dear students, now I also the last stage from the karyokinesis and the cytokinesis. Here we also know about the one cell is divided into the two cells. The previous cell is called the mother cell, and when they are divided, we also tell the daughter cells. Here the students, please everyone draw this 5 phase of the karyokinesis. Thank you.